The secretion is the third process of renal exchange. And just to remind ourselves, the big picture here is that we're fine-tuning this filtrate. So reabsorption is taking things that we wanted from the filtrate and moving them back into the blood. Secretion is going to be the exact opposite. It's going to be taking things that we no longer want, that are still in the blood, and moving them back into the tubular fluid so that they can be excreted from the body. The barriers for secretion are going to be the exact same barriers for reabsorption. We're just moving in the opposite direction here. And the substances that are secreted are going to be a little bit different, and they're going to be secreted in different areas of the tubular structures. So in general, or generally, potassium is secreted. Hydrogen ions are secreted as a part of our acid-base balance. Choline is secreted, and choline is removed from the blood and back into the tubular structures. Creatinine is also secreted. I'll talk a little bit about creatinine clearance. And then things like penicillin and other drugs are secreted as a way to remove them from the body and to help detoxification of some of these drugs from the body. Okay. All right, so that brings us to a few checkpoint questions, and I won't go over these here, but I will ask you guys to write down a descriptive response to these questions, and then I'm going to create a question in our essay practice questions folder in Blackboard, and you can post your response to this first checkpoint question there. So stay tuned for that question in Blackboard, and I'll send out an announcement when that goes live. Okay, so as an extra credit question, you're going to create an essay response to these checkpoint prompts and submit it in Blackboard. Okay, let's see, I got another question here. Okay, so someone asked me to repeat the barriers to absorption. Really good point. So I'll go back here, and we'll talk about the barriers to reabsorption, which are the same barriers to secretion. So the barriers are going to be the epithelial cell. Remember that the tubular structure here is lined by an epithelial cell. The second part of that is going to be the basement membrane of that epithelial cell. We've also got the peritubular space and then the endothelial cell, okay? So actually, let's go back to this image. So we've got the lumen of the tubular fluid and the lumen of the peritubular capillary. And so in order to move in either direction here, there are certain barriers. So the first one is going to be the epithelial cell, the basement membrane of the epithelial cell, the peritubular space, and then the endothelial cell of the peritubular capillary. And then we can get into the plasma, or if we're going in the opposite direction, the membrane structures are going to be the same, but we're just moving from one direction to the other. So this is the general direction for reabsorption from tubular fluid to plasma. And then the general direction for secretion is going to be from plasma to tubular fluid. Okay. All right, let's see if there are any other questions before we continue. Okay, next. All right, so what we're going to move on to speak about now are the regional specialization of the renal tubule. And I was alluding to this earlier when we drew out the nephron, right? So each area of the nephron is actually responsible for a different function in terms of fine-tuning this filtrate that becomes urine. So far we looked at what happened in the proximal convoluted tubule, so this part of the tubule. And then now we'll look at what happens at other areas of the tubule, and we'll speak in general about different functions of different areas of the tubular structures. So there, first of all, there is non-regulated versus regulated reabsorption. Then I'll kind of describe what the regulation versus non-regulation means, and then I'll talk about what areas have regulated and what areas are non-regulated. So when we talk about regulation, we talk about things that are being influenced based upon GFR or hormone release or sympathetic versus parasympathetic involvement. And so we can talk about non-regulated reabsorption, which mainly happens in the proximal convoluted tubule. So this is not regulated. There aren't any hormones that can change how things are reabsorbed in the proximal convoluted tubule. There aren't any neurotransmitters. There is not any influence based upon GFR. All of the substances that are reabsorbed in the proximal convoluted tubule are freely reabsorbed and are not regulated by any external factors. Reabsorption and secretion that takes place in the distal convoluted tubule, as well as the collecting duct, is very heavily regulated. And again, this means that there are external factors that influence the rate of reabsorption at these parts of the tubule. Things like hormones. We're going to speak about aquaporin, ADH, or vasopressin, which influences how reabsorption takes place in the distal part of the tubule. The autonomic nervous system also has an influence on the distal parts of the tubule. So these areas of the tubule, the distal convoluted tubule, which is this area, and the collecting duct, which is this area, the final part of the tubule, are very heavily regulated by external factors. And then we've got the loop of Henle, which is a little bit different because the loop of Henle is really involved in water conservation. And I'll talk about how the loops of Henle of different animals vary depending on their need to conserve water. This is actually a very important evolutionary feature of different species depending on their need to conserve water or to go for extended periods without water, okay? So proximal convoluted tubule, non-regulated reabsorption, distal convoluted tubule and collecting duct, regulated reabsorption and secretion, and then loop of Henle is really for water conservation. Okay. This is another really nice table that kind of summarizes a few important concepts here. And I don't want you to memorize this per se, but have a good understanding of a few important trends. So the first important trend is that the proximal convoluted tubule is where we have most of the valuable, nutritive, important substances being reabsorbed. Things like glucose, things like amino acids, vitamins, urea, choline. Choline is going to be really important for synthesizing one of our most abundant neurotransmitters, acetylcholine. So these are vital substances that are reabsorbed at the first part of the tubule, the proximal convoluted tubule, okay? So that's sort of the kind of first trend that we see. The second trend is that water is reabsorbed everywhere except the ascending limb of the loop of Henle. So we've got water reabsorption at the proximal tubule, water reabsorption at the descending limb of the loop of Henle, water reabsorption in the distal tubule, and the collecting duct. And this really speaks to how important it is to reabsorb water. So water reabsorption takes place everywhere except for this ascending limb. And then the third trend is hydrogen ions. So hydrogen ions are usually secreted, and this happens, again, very prevalent throughout different parts of the tubule. They're secreted in the proximal tubule, secreted in the distal tubule, secreted in the collecting duct. And this is going to make more sense when we look at the function of the kidneys in terms of water, and not water, in terms of acid-base balance. So the ability for the kidneys to help with us balancing the acidity and the alkalinity of our blood has to do a lot with the fact that hydrogen ions are secreted at the proximal tubule, the distal tubule, and the collecting duct. Okay, all right. So let's go ahead and look at the non-regulated reabsorption in the proximal tubule a little bit closer, and this will bring us to describe the anatomy of the proximal convoluted tubule cells. So this is where most of the reabsorption takes place. So 70% of the sodium and water, and then 100% of the glucose is reabsorbed across the proximal tubule. And this happens via this structure that's called the brush border. So the brush border is this highly folded membrane of the apical surface of these epithelial cells of the proximal convoluted tubule. That was a lot of terms. So the apical surface of these cells has the microvilli, which are these apical modifications, which allows the surface to be really highly folded. And we know that anytime we see this highly folded nature of the membrane, this is an adaptation which increases surface area, which increases reabsorption, okay? And so because we see this highly folded apical membrane, we know that reabsorption is a really important function that takes place across this membrane. Another adaptation of the proximal tubule cells is that they have leaky tight junctions. So in between one cell and the other, there is a leaky tight junction, which allows for some degree of paracellular transport. And what that means is that this cell can communicate with this cell, and this cell can communicate with this cell through the leaky nature of these paracellular or tight junctions. And that is also indicating to us that this area of the tubule is not as heavily regulated. There can be free movement of solutes, free movement of water, and there can be some level of paracellular transport or transport in between cells themselves, okay? And this is a closer view of those modifications. So the microvilli, which form this brush border, and as you can see, we get the term brush border because if you look at a histological representation of this, the apical surface of this membrane, it appears as the bristles of a brush, right? This highly folded nature of the apical surface of these epithelial cells. And then the second modification here was the leaky tight junctions, which allowed for some paracellular transport. So not only transport in between cells, but things can move in between these two cells. So certain solutes and particles can go from the lumen here in between the sides of these cells and make their way across into the blood. Okay, that's what we mean by leaky tight junctions. Now let's look at the regulated reabsorption and secretion that happen in the distal tubules and in the collecting duct. So here we see different modifications. So we do not see the brush border, we do not see microvilli, and we do not see leaky tight junctions. Because this part of the tubule, because transport here is heavily regulated, we see very tight junctions that are very well secured. And so there can be no paracellular transport, or there is limited paracellular transport. So we see that there is a limited movement between the lumen and the underlying peritubular capillary, and there's also limited movement from one cell to the other, okay? And then there is no brush border that is indicating that regulated nature of reabsorption. All right. And then the third modification, or the third region of the tubule that had its own modification was the loop of Henle. And the loop of Henle, as I mentioned earlier, helps to establish conditions that are necessary to concentrate or to conserve water or to concentrate the urine. And this is what allows us to minimize water loss. So this is actually a really cool evolutionary modification. And so in humans, because we have this long loop of Henle, we talked about the fact that the loop of Henle's extend from the cortex, which is this region, all the way down into the medulla, which is where the loop actually dips. And because humans have this long loop of Henle, this is an evolutionary modification that allows us to be able to go for extended periods of time without water. So we can conserve or hold on to water, and we can minimize our water loss, which is a protective measure, right? Especially humans who live in areas where there's more of a desert environment, they are going to exhibit longer loops of Henle's, which is going to facilitate that ability to conserve water and to concentrate the urine and minimize water loss. If we look at other animals like camels or desert rats, they have really long loops of Henle's. And that tells us the ability of these animals to go for even longer, much longer periods than humans can endure, for even longer periods without water because of their really amazing ability to concentrate their urine, where they're really only losing waste. They're holding on to all the water that is possible, losing absolutely only what is wasteful or what is not needed by the body, and minimizing how much water they're losing unnecessarily.